overhead for as long as it works, but I have a spare. Ready.
That's because you use it too much. I remember you went to like 5 of these things last
I have already gone through 3, and I've only been teaching 5 years, so that is. Oh, by the way, I have a couple things going on. I want you guys, I do need your guys' help. If you haven't noticed, there's 5 onions sitting, well, 6 if you count the green ones, sitting in just some beakers of water. I am trying to get the roots to grow again, okay? Why? Well, I'll explain as we go further along, but I need you guys to kind of make some observations every day. And now you see some roots hanging down, but they've got to be new ones that are actually living. So I want them to grow for about 1 centimeter, and then we'll go to the next step. But that's for a lab that we might do as well. So I knew somebody was bound to ask, why are there onions in beakers of water up here? So that's a lab that's coming up.
To make my eyes water.
I don't think it would, because they're not cut. So I don't think it's going to start stinking up the lab or anything. But if I could, just maybe I'll do that just to torture you.
I'll move.
Take him on his cheeks.
Yeah. Okay. All right. I'm not sure. No, we didn't get that far. But I want you to look up here and tell me, is that a genotype or a phenotype?
Genotype.
All right, then it is a genotype. Okay, remember, genotype is a code. It's what's written in your DNA. It's your genetic code. Now, obviously, we use symbols to represent that code. That's why you have big letters and little letters.
Big letters are dominant.
The big letters are dominant. The little letters are recessive. Okay.
I just nailed that.
Good. That is good. Remember, you should also know what the word allele means. An allele is like the most. Its technical definition is the different form of a gene. Each of these represents a gene. Okay, so little b is representing a genotype, or what a certain gene is saying. Okay, maybe it's for eye color, so this is the gene for eye color. An allele means there's an alternate version for that gene. And you can see that there are actually 2 letters here. Each of those letters is an allele. That's an allele. That's an allele. Now, this particular genotype has 2 of the same allele. The alleles are exactly the same. But the one in the middle here, it's different. One's capital letter, one's lowercase. Okay, so the alleles are slightly different. Over here, the alleles are the same. Okay. I can't remember if I said this, but if I did, I'm just going to repeat it, but I don't think I did. Did I draw a picture of a cell?
Yeah.
What was I trying? Yeah, that's right, I did. Okay. Then I'll just remind you, I won't redraw it. The last thing in your notes, I believe I drew a picture of a cell with 6 chromosomes. I won't recopy it, but I drew one with 6 chromosomes. And I said, okay, last chapter, we didn't distinguish between those 6 chromosomes. They were 6 chromosomes. Remember, in mitosis, you just needed to make copies of each of those 6 chromosomes, and good stop. You needed to make copies of those 6 chromosomes and send them into each of new cells. But now it matters. And I think the last thing I got done telling you was that they have a pair. They have a match. What did I name those pairs? 2 chromosomes that hook up and find their partner. Maybe I didn't give you a name for that. Let me quickly write this out. Just so we're on the right track. 6 chromosomes. What's going to happen, those 6 chromosomes actually have a buddy. We didn't care about their buddies last year, or last chapter. Now we do. 1, 2, 3, 4, 5, 6. I still have 6 chromosomes, but now they're matched up with their buddy. And these buddies, are called, the 2 of them together are called homologous chromosomes. That's a big word, homologous chromosomes. I know it's a big word, but it just basically means a chromosome and its pair. Well, why is it its pair? Now, this has to do with those alleles we just talked about. Because, well, let's use this one as an example. I'm going to take this genotype, has two different alleles, and I'm going to stick those alleles on the chromosomes. Homologous chromosomes are chromosomes that basically code for the same traits, but they could have alternate forms of those genes there. This big blue chromosome has contains the information for that particular trait. Maybe the B, the big B, stands for brown eyes. The little b stands for blue eyes. So this pair of homologous chromosomes at the very top has one allele for that trait. Okay, keep that in the back of your mind. I'm going to keep going here. All right. Let's go back to Mendel. Mendel came up with some laws. 3 of them I want to talk about today. That's my goal of today, is to talk about Mendel's 3 laws. One of them is called the law of dominance, which actually, you guys, this is the easiest of the 3. They get a little harder as you go along. Some alleles are dominant. Some alleles are recessive. So I really want you to understand that word dominant and recessive. We talked about them already, but now I want to make sure you really understand them. If something is a dominant, if something is dominant, I like this word, masks. If there is a dominant, let me kind of write this, a dominant allele masks the presence of another allele. Okay. Recessive. A recessive allele, more specific. A recessive allele will only be expressed. You'll only be able to see it, in other words, if no dominant allele is present. Recessive allele is shown or seen, expressed, if no other dominant allele is present. All right. I'm writing the 3 different genotypes here again. Here's a genotype, here's a genotype, here's a genotype. Each genotype has 2 alleles. Let's say the big one, the big allele stands for dimples. The little d stands for no dimples. Who has dimples in this class? Anybody? You're the only one that has dimples? Oh, okay. Okay, there should be a few of you. Not a lot of people have dimples, which is strange because I wrote it as the capital letter, but that's right. Believe it or not, dimples is it's a dominant trait, but you don't see it very much in the human species. It's kind of funny the way it works that way. Yeah.
How come my sister only has one?
I have one too.
Oh, you know what? It still is. That's a good question. I don't know. Yeah.
I have a big one.
That's funny. Somebody in the other class only had one dimple too. I think it was Mackenzie. Swearing. Kind of funny. Okay, here's what I'm getting. Dominant, recessive. Hey, in this first genotype, you see 2 dominant alleles. Obviously, is this individual going to have dimples or not?
Yes.
Yes. Middle one.
Yes.
Yes, they're going to have dimples. Even though they have the recessive allele, it's being masked by the dominant allele. That's what I mean by the word mask. So even though this individual has a recessive allele, lowercase letter, they're not going to express that trait. They're going to express the dominant trait. They're going to express dimples. Only when you have 2 lowercase letters will that recessive allele be expressed. So this individual over here, no dimples. Very important that we get that. Okay, that's all that says. Oh, by the way, I ran out of room.
Can't step right up anymore. So wait, this projector's gonna go out soon or something?
No.
Oh, I said it was.
It might. Want you to know the difference between a homozygous genotype and a heterozygous genotype. All right, going back to the examples I just put up there for dimples. The word homo, the prefix homo means same. The word hetero, the prefix hetero means different. If you have two letters that are the same, that genotype is going to be considered homozygous. Well, looking up here, here's a homozygous genotype. The letters are the same. But look over here too. The little d and little d, they're the same. So that is considered a homozygous genotype as well. You can differentiate. This is a homozygous dominant genotype. Obviously, this would be considered a homozygous what? Recessive genotype. On tests, I may very well have a question where I say, okay, let's say T's are the, big T represents a tall allele, little t represents a short allele. What would be the genotype of a plant that is homozygous dominant for height? And you would write down big T, big T. Okay, so make sure you understand if I say homozygous dominant, you automatically think of two capital letters. If I say homozygous recessive, you automatically think of two lowercase letters. Heterozygous, they're different. You can't be heterozygous dominant or heterozygous recessive because heterozygous means it's different. So that middle genotype is considered heterozygous. So on a test, I could say, Mom is heterozygous for eye color. That automatically means you're going to have one capital letter and one lowercase letter used to represent Mom's genotype. So get used to those words real well. All right. Mendel's second law. Mendel's law of segregation. And if you know what segregation means from like a history class, you would know segregation means to separate. Well, that's exactly what this law is stating. I'll write it down and then I'll show you a picture. Parents pass only one allele for a trait to their offspring. Okay, well, let me draw a picture. Pictures are better. For simplicity's sake, I will draw just one pair of homologous chromosomes. I'm not going to draw a lot of them. So let's say I have a female cell here. And I've got a pair of homologous chromosomes. All right, these chromosomes are going to, I want to talk about this gene location right here. That gene location, we'll make it for dimples. Let's say the female has a big D at one allele. One allele is big D and one allele is small d. So obviously this female has dimples. Okay. All right. What this law is saying is that when this female goes on to make eggs, you know, females make eggs, males make sperm, but when this female goes on to make eggs, those alleles are going to separate. So one egg might get the big D, and the other egg might get the little d, but they will never go together. They will never go together in one egg. Each egg is only going to get one or the other. That's exactly what that law is saying. So parents pass only one allele for each trait to their offspring. What?
She looked at me and smiled when you said big D, little D.
Oh. I was going to say this isn't all that exciting. I didn't think you. Okay.
No, it's not. But I don't know why you looked at me.
All right, guys. Here's why you. Let's do this. Let's try another circle. Let's make this a male cell, and let's do the same thing. Let's give him 2 chromosomes. They're homologous. And let's look at his alleles for dimples. And the boy's going to be different. He's not going to have one big D, one little d. He's going to be homozygous recessive for dimples, which means he doesn't have. When this male goes on to make sperm, this law is saying that each sperm is only going to receive one or the other. Now, in this case, he can donate nothing other than a lowercase d. But here's why it matters. What would happen if this egg hooked up with this sperm to produce baby? That offspring, that baby, will or will not have dimples?
No.
Will not have dimples. Mom did. Mom had dimples. Dad did not. Baby may or may not, but in this case, it does not. This is one reason you don't look like your parents, because the alleles for each trait are going to go separate into separate eggs and sperm. It is going to be luck of the draw with which one you get, which allele you get. It just happens. It just has to happen on which egg actually hits what meets up with what sperm. Okay, Mendel's law of segregation, pretty easy. Now we're getting a little detail. Oops, that's a dirty notepad. All right. Mendel's last law of, and it's a big 2 words, independent assortment. Okay, Mendel's law of independent assortment. Oh, well, independent means you're fine on your own. Assortment, how you sort yourself. This law only applies when you're talking about 2 or more traits. Now, in the last example, I was only talking about 1 trait, dimples. This law only applies if you're talking about more than 1 trait. In other words, maybe dimples and eye color. So you got to be talking about more than 1 trait when it comes to this law. Here's what the law says in written form. The alleles for 1 trait, dimples for example, have no effect on the inheritance of another trait. In other words, what this law says is just because you have dimples doesn't mean you're going to have brown hair. Or just because you have dimples doesn't mean you're automatically going to have blonde hair. In other words, the traits act independently of each other. The alleles for those traits act independently of each other. Okay, here's what I want. Here's what I want you guys to be able to do, and this will set yourself up for knowing how to do a Punnett square. I've got a genotype here. There's 2 traits represented by this genotype. There's 4 letters, yeah, but remember, each trait is controlled by 2 alleles, so that's why you have 4 letters. So this represents 1 trait, this represents another trait. I'll tell you what, for simplicity's sake, we can maybe add real life. No, we're not going to confuse it, okay? There's a genotype. Okay. This law is saying that when this individual with this genotype goes on to make gametes, by the way, just remember, gametes are nothing more than a fancy way of saying a sex cell, which, for example, would be like a sperm or an egg. So females, ladies, the type of gametes you make are eggs, and males, the type of gametes you make are sperm is what that means. But what I'm saying here is when this individual goes to make their gametes, when it comes to what combinations they can make, it doesn't matter about the other one. So just because, in other words, just because this particular gamete's going to get a capital A doesn't automatically mean it's only going to get the capital B. It could very well get the lowercase b too. Yeah, Jerry.
What's the actual name for like the little four? Oh, Punnett squares.
Yes.
Yeah.
Yep. We'll be there in like 2 minutes. Yep. This will set yourself up for it. Good.
Good job, Jerry.
In other words, this law is saying that capital A could go with a capital B, or the capital A could go with a lowercase b, and it has an equal chance of being one or the other. This law also says the gametes can have a lowercase a with a big B, and it can also have a lowercase a with a little b. In other words, what I just did here, I came up with all the possible combination of gametes that this individual can make. Now, there's only 4 in this case because I'm only talking about 2 traits here. Keep in mind, humans, for example, have like 50 or 60,000 traits. That would be a lot of circles and a lot of different combinations. Okay. Punnett squares. No. If you understood what I just said, you'd have no problem with Punnett squares. By the way, a Punnett square is a tool used to predict, and I'm going to underline the word predict, to predict an offspring's genotype and phenotype. Here's what that means. Doctors use these things all the time. If you have a history of a genetic disorder in your family, and let's say you're going to get married, and your spouse wants to have kids, but you're worried. You're like, ooh, I have this genetic disorder that kind of runs in my family. I don't know if my kids have a chance of getting that or not. You would go to a genetic counselor, a doctor, who would do a Punnett square or something very similar to a Punnett square and be able to give you an idea of what chance you have of having a kid that has that disorder. They'll literally say, no, you know what? You have a 25% chance of your kid inheriting that disorder. Or they may say you have a 75% chance of your kid inheriting that disorder. They basically can figure out your chances of producing an offspring with that genetic disorder. So it's actually used all the time. So here's how they work. I'll start off really, really easy with one trait. Let's say I have, let's just do one trait, so it's only going to be 2 letters each. Let's say the male has a genotype of that, and the female has the same thing. Okay. Let's say the big B stands for the ability to roll your tongue. Believe it or not, that's a genetic trait. You guys know what I mean by that? If you can make your tongue into roll it up, that basically means it's a genetic trait. There's actually not everybody can do it. But let's say the capital B means that you can roll your tongue, is the allele for the ability to roll your tongue. Little b, you cannot roll your tongue, but not roll.
What if you just touch your nose with your tongue? Is that one too?
I don't think so. I know rolling your tongue is, but I don't think that is.
I can't do it.
Okay. Other traits, guys, by the way, guys, there's a lot of traits that you guys are going to be looking at in the lab tomorrow. You're going to be looking at yourself and the traits you have, whether or not you have a widow's peak, which would be like an X, or if your hair comes down to a point in the front of your forehead.
Your ears.
Another attached or unattached earlobes. Like I definitely have attached earlobes because there's no little flap at all. These are all genetic traits that you guys are going to be looking at tomorrow. But let's say I want to predict. Okay, let's say I know my genotype is this. I know, the husband's genotype is this, and I want to figure out what chances do my kids have of being able to roll their tongue.
50%.
Okay, no. Here's what you do. Since this is only a one trait cross, you have to you make a very simple 4-square Punnett square. This is the hard part. This is the part that everybody seems to not know what to do. The hardest part about doing a Punnett square is figuring out what letters you put outside the box. To do that, you have to take the male and basically you have to take them separately. First you start with the female or male, then you do the other one. In other words, I'm going to start with the male here. The male's genotype is big B, little b. Now you have to remember what we said about those alleles when you make gametes are going to separate. What you're putting over here outside the box are all the possible gametes that this individual can make. So in this case, the male is big B or little b. When this male goes to make sperm, those alleles are going to separate, and that is what I would place above the boxes. Only one letter. Why only one letter? Because each of those sperm is only going to receive one allele, one letter. Female, same thing. When she goes to make her eggs, those alleles are going to separate and go into individual eggs. That's why I split them up. Now the fun part. You got to figure out what's going to go inside the boxes. What's going to go inside the boxes are what all the kids might look like. There's going to be 2 letters for each box. You have to match up the letters. Box number 1, upper left-hand corner here. You got to bring down this letter and bring over this letter. Big B, big B. Over here, you're bringing down this letter with this letter. Big B, little b. It down over here, lower the third box, 1, 2, lower left-hand box. You're going to take the big B and hook it up with the little b. Again, I'm dragging this letter right here with this letter over here where they match up and hook up to the letters you draw there. This last box off to the bottom right hand, I'm going to take whatever letters. I'm going to take this letter with this letter, little b, little b. Okay, does everybody see how I filled in the boxes? That is very important. What I did was I determined these 2 individuals, female, male, whatever, inside the boxes are all the possible combinations of offspring that they can have. 1 box is big B, big B. 1 box is big B, little b. Another box is big B, little b. And 1 box is little b, little b. From this information, I want you to tell me what percent will have the ability to roll their tongue. 75%. Why? How did I get that? 3 out of the 4 boxes are going to have kids that are going to have they have at least 1 capital letter. If they have at least 1 capital letter, they're going to be able to roll their tongue. 1 out of the 4 boxes has 2 little b's. Those individuals will not be able to roll their tongue. 25%. 1 out of 4 is 25%.
So it doesn't matter if it's like in the bottom left box. It doesn't matter if you have like little b, big B.
No. Good question. The question was this. Would it have mattered if I wrote it big B, little b, or little b, big B? No, not at all. I'm just always in the habit of writing, if there is a big uppercase letter, write that first. But it does not make it wrong if you in this bottom box put little b, big B. Very good question. That's an excellent question. It still means the same thing. Okay, by the way, hopefully you see I have, 1/4. Let's do it this way. I also want you to be able to give me both a phenotypic ratio, don't let that word ratio throw you off, and a genotypic ratio. In other words, remember what phenotype is. Phenotype is what you see. All this is saying is, okay, what are going to be your ratio of people who can roll their tongue and not roll their tongue?
4 to 1.
3 to 1.
3 to 1.
How did I get that? Because 3 of the 4 boxes can roll their tongue. 1 out of the 4 boxes cannot roll their tongue.
How do you know
Phenotypic ratio 3 to 1. Huh?
How do you know phenotypic though?
I-
Yeah, you can see it, but what is how do you get the genotypic? Like,
Genotypic ratio, we're going to do that next. Okay, genotypic ratio. This is where you have to differentiate between those folks that are homozygous dominant and the folks that are heterozygous. All of them are going to be able to roll their tongue, but their genotypes are not the same. The genotypic ratio for this one would be 1 to 2 to 1. One individual is big B, big B. The 2 here stands for the individuals that are heterozygous. 2 of the 4 boxes are. And this last number over here represents the genotype of those that are little b, little b. By the way, let me go back up here and tell you where I got this 3 to 1. The 3 comes from the basically the number of people that can roll their tongue, or the number of boxes where that represent kids, individuals that can roll their tongue. That 1 not roll their tongue. Is everybody with me so far? Is there anybody who is lost? Because it gets a little harder. Just, do you want me to repeat this in any way? Okay, let's do one more. I want to see if you know what you're doing. The big T allele stands for tall. The little T allele stands for short. Let's talk about plants now, because we don't want to forget our, members of the plant species kind. I want you to cross a homozygous recessive male with a heterozygous female. I want you guys to, number 1, write out their genotypes. Number 2, do a Punnett square. And number 3, tell me the phenotypic and genotypic ratio. In other words, I want you to do exactly what we just did with rolling your tongue, and do it that here. If you understood what I said, this shouldn't be a problem. Step 1. Homozygous recessive. Well, what's the genotype going to be? No, homozygous recessive. Homo same, recessive little t, little t. What's my heterozygous genotype going to be?
Big T.
Big T and a little t. Okay, so step 1. I got my genotypes. Now I'm going to make up a Punnett square. Simple 4-box one, because I'm only talking about one trait. Let's take the male here. Little t, little t. Remember, I am just listing the possible gametes outside the box. Well, those T's are going to separate according to the law of segregation. That's why there's only going to be 1 in each. This male is going to only be able to make sperm that have the little t in them, but each sperm's only going to have 1 little t. The female over here, I'm going to split up her alleles as well, according to the law of segregation. Put her alleles down there. Cross them. Big T, little t. Big T, little t. Little t, little t, little t, little t. Everybody following me so far? What percentage of those plants are going to be tall?
50%.
50%. Right here and right here, those two boxes. Because both of those plants are going to be tall. The only way those plants are going to be short is if they have two lowercase letters. So 50% will be tall, 50% will be short. The genotypic and the phenotypic ratio are both going to be 1 to 1. Or 2 to 2. You could have put 2 to 2. 2 are that way, 2 are the other. So the genotypic and phenotypic ratio are the same. Now, gone. How many? Raise your hand if you consider this easy so far. Raise your hand if you're like, I'm getting there, but I think it's okay. All right. You guys are. All right, here. I'm going to leave you with one final one to do. We have plenty of time. We're not doing one trade anymore. We're doing 2. I even made the Punnett square for you. All right. I want you guys to, well, first step is figure out what letters are going to go outside the box for both Mom and Dad. Then, you know, there'll be plenty of time. You can probably fill in the boxes. So try that. I'm not even going to give you the answer, but I am going to kind of sneak around the room and see what you're doing. Did you need the... That one? Okay. Okay, let me give you some hints if you're stuck. You will only have 2 letters above each box. 2 letters here. One of them better be an R and one of them better be a Y. Do not put 2 R's together. Do not put 2 Y's together. That's a common mistake. Remember, alleles will segregate. This egg that Mom makes cannot have both a big R and a little r. It's got to be one or the other. But that R is going to hook up with one of the Y's. Let me get you started here. Let's take Mom's. Actually, both of the genotypes are the same, so it doesn't matter. But I'll take Mom's, put her along the top here. Mom can make an egg with a big R and with a big Y. Mom can also have the ability to make big R, little y.
And then big Y, little Y.
I'll let you figure out the rest. Does that get you started?
My chair's a villain.